Hello, everyone. Welcome to our lecture on our medical biochemistry series on signal transduction. I am Dr. Muzamil Liaqat Ali, Assistant Professor of Biochemistry. Today, we begin one of the most clinically relevant and high-yield topics in the entire subject: how cells receive, interpret, and respond to external signals. We divide this lecture or this session into two parts. In part one, we will master the fundamentals, the basics of a signal transduction, the GPCR structure, and the classic CAMP signaling pathway. So let's proceed with the clarity and precision. As medical students, you must achieve these objectives today. The objectives will be the following: describe the general features of the chemical messengers and receptors, differentiate between endocrine, paracrine, and autocrine signaling, explain receptor specificity and its clinical importance, draw and label the structure of a typical G-protein coupled receptor, the GPCR, list the major components of signal transduction pathways, classify signaling molecules, the ligands, on the basis of solubility and receptor location, distinguish between enzyme-linked, ion channel-linked, and G-protein cell surface receptors. And finally, you need to explain the step-by-step mechanism of the signal transduction pathway, that is Gs-cAMP-Protein Kinase A pathway and its termination, right? So let's begin with the general features of the chemical messengers and receptors. A chemical messenger, whether a hormone, cytokine, neurotransmitter, or a growth factor, is secreted by specific cell in response to a precise stimulus. It diffuses or is transported via blood or extracellular fluid to its target cell. There it bounds to a highly specific receptor located either on the plasma membrane or inside the cell. This binding event triggers a cascade of intracellular reactions that ultimately produce a biological response. Importantly, every signal must be terminated. Failure to termination leads to uncontrolled cellular activity. So now let's move to the common modes used for transmission of regulatory signals between cells. We classify signaling by distance, right? The very first one is endocrine. In this type of signaling, the messengers travel long distances via blood. For example, insulin from pancreas to distant target tissues, right? The next one is the paracrine. Messengers act on immediately adjacent cells. For example, the neurotransmitters at synapses, right? The next one is the autocrine. The cell secretes a messenger that binds back to its own receptors, common in immune regulation and cancer progression, right? So, master these distinctions, right? They explain how the body coordinates systemic versus local responses. Now, move to the specificity of receptors. Receptor specificity is the hallmark of accurate signaling. Each receptor binds only one chemical messenger for or a very close or very closely related group. Each receptor activates a unique downstream pathway. Only target cells express the appropriate receptor. Clinically, loss of specificity or a failure to terminate the signal contributes to a disease such as cancer, hyperthyroidism, or many endocrine disorders, right? This is why most modern drugs are designed as receptors or receptor agonists, antagonists, or modulators, right? Now, let's move to the structure of a typical C-protein coupled receptor, that is GPCR, of the plasma membrane. As you can see on your screen, A typical GPCR is an integral membrane protein with a 7 alpha helical transmembrane domains. The N terminus is extracellular and forms a ligand binding pocket. The intracellular loops and the C terminus interacts with the heterotrimeric G proteins. This 7 transmembrane architecture is conserved across a large family of receptors in the human body, involved in vision, smell, taste, and the action of more than 50% of all prescription drugs, right? Next is the components of the signal transduction. The complete pathway requires a chemical messenger, the ligand, right? The most important thing, the stimulus, the chemical messenger. Next is the receptor, right? Receptor, the G proteins, the effector enzymes. For example, the adenylyl cyclase, phospholipase, C-guanylyl cyclase. The next is the C messengers, right? Second messenger can be or maybe the CAMP, CGMP, IP3, that is inositol triphosphate, DAG, that is diacylglycerol, or the calcium or colmodulin, and protein kinase. For example, protein kinase A, protein kinase C, and the protein kinase G, right? We will master each component by today, right? Now, let's move to the adenosine monophosphate. The ATP is converted to cyclic AMP or the CAMP by an enzyme adenylyl cyclase. CAMP is the classic second messenger that amplifies the original signal inside the cell. I will show you exactly how this conversion occurs and why it is so powerful, right? Next is the guanosine monophosphate and CGMP. Similarly, the GTP is converted to CGMP by guanylyl cyclase. We will study CGMP in detail in part 2, right? But note its structural and functional similarity to CAMP, that is cyclic AMP. Now let's move to the classification of cell signaling based on the distance, right? The very first one is the endocrine signaling. In endocrine signaling, endocrine, the insulin or the paracrine, the neurotransmitters, or the autocrine, the growth factor in tumors. This classification is essential for understanding hormone physiology, right? This is what you have already gone through. Let's talk about the signaling molecule, the ligands, its classification, right? Ligands are classified by solubility and receptor location, right? The very first one is the lipophilic hormones with the intracellular receptors. For example, the steroid hormones, thyroxine, vitamin D, and retinoic acid, right? The next one is the lipophilic hormones with the cell surface receptors, right? For example, the prostaglandins. The next one is the hydrophilic hormones with the cell surface receptors. Peptide hormones like insulin, the biogenic amines like the epinephrine. This classification determines whether the hormone acts via gene transcription or the rapid second messenger pathways. Next is the cell surface receptors. Let's talk about cell surface receptors. Hydrophilic ligands binds to the membrane receptors, causing conformational change that initiates a signal transduction. We recognize three major classes. The very first one is the enzyme linked. For example, insulin receptor with the tyrosine kinase activity. The next one is the ion channel linked. For example, nicotinic acetylcholine receptor at the neuromuscular junction, right? The next one is the G protein linked, the largest family, that is GPCRs, right? G-protein coupled receptors. Now, a question arises, right? Why these proteins are named as G proteins? G proteins bind guanine nucleotides, GTP or the GDP. Ligand binding to the receptor catalyzes GDP to GTP, right? Exchange on the alpha subunit. The GA, GTP dissociates and becomes the active form that modulates effector enzymes. Signal transduction via the GS or stimulatory G protein via alpha protein or via the CAMP as a second messenger. As you can see, an overview with this, right? First, you need to have a ligand, the very first messenger, that goes to the GPCR, then the GS protein, then it moves to the adenylyl cyclase, then the CAMP or the second messenger come into play. The next is the protein kinase A, and finally you get the cellular response. So these are the steps, right? Steps of CAMP pathway, detailed mechanism. Walk through each number, step slowly with clear diagrams, right? So ligand binds to the GPCR, that promotes a conformational change, right? Next is the receptor activates the GS or the stimulatory protein to GDP, that replaced by the GTP on the GA or the subunit of the G protein, right? G alpha. The GDP, that is basically the combo, right? That dissociates and activates the adenylyl cyclase. Adenylyl cyclase converts ATP to CAMP and the PPI, right? Pyrophosphate, right? So next is the CAMP, that is being, produced by the conversion of the ATP to CAMP by the adenylyl cyclase. The CAMP activates the protein kinase A, the pKa, right? Next is the GTPase activity by the GA hydrolyzes GTP to GDP, and ultimately that leads to the inactivation of the cascade, right? So receptor is phosphorylated by G protein coupled receptor kinase activity and binds a certain, right? That is the desensitization, right? This is what you have, an illustration from the Lippin coil. So this is what that outlines the different types of signal transductions, right? Right? So actions of CAMP on the protein kinase A. Let's discuss, some of the points related to it, right? CAMP binds regulatory subunits of the PK, releasing catalytic subunits that phosphorylate target proteins, producing the final cellular response. The glycogen breakdown or the gene transcription are the examples, right? So as you can see on your screen, these are the steps of the CAMP pathway. These are the actions of the CAMP on the protein kinase, right? This is what we have already discussed, right, in our previous slides, right? Now, the termination of the response. Signal termination occurs via the intrinsic GTPase activity via G alpha, right? Phosphodiesterase that hydrolyzes the CAMP to AMP. And the protein phosphatases that removes phosphate groups, right? And along with that, the fourth mechanism that leads to the signal termination via the receptor phosphorylation or the ascertain binding rate. Thank you very much. We have completed the part 1 of this lectures on the signal transduction. So you now fully understand the fundamentals, the basics of the signal transduction and the complete CAMP pathway. In part 2, we will cover the CGMP, IP3 or the DAG pathway, that is inositol 3-phosphate or the diacylglycerol pathway, the intercellular receptors for steroid and the thyroid hormones, and important clinical correlation. Right? Thank you for watching this lecture. Please like, share, and subscribe. Turn on the notification for the part 2. Write any questions or comments about the GPCR or the CAMP in the comments, right? I'll reply personally, right? This is Dr. Muzamil Liaqat Ali signing off from the part 1. Revise the CAMP steps by drawing these before watching part 2. See you soon. Thank you very much.